Welcome, everybody. Welcome to our Life Science Cafe for this month. My name is Elise McCormick, and I am a PhD student in the Organismal and Evolutionary Biology program at UMass Amherst. And I am your MC for tonight's cafe. Just to give you a little bit of background on our group, Life Science Cafe was established in 2011 and is organized by graduate student researchers at the University of Massachusetts Amherst. Each month, we invite speakers to share and discuss their research. We would like to thank our funding sources, the Organismal and Evolutionary Biology program, the Graduate Student Senate, and the Graduate School at UMass, the National Science Foundation, and our generous UMass Gives donors. If you'd like, you can subscribe to our mailing list and stay tuned for our future cafes. We're also now hosting our events hybrid to benefit both of our virtual and in-person viewers. In addition, we're also trying to make our events more accessible, which includes having closed captioning and events in other languages. If there's anything that you feel would improve our accessibility of events, please feel free to let us know. So with that, today I have the pleasure of introducing Dr. Kristen DeAngelis. Dr. DeAngelis is a professor at UMass Amherst. She received her bachelor's degree from Harvard University and her PhD from UC Berkeley. She then went on to do postdoctoral work at the Lawrence Berkeley National Laboratory, as well as the Joint Bioenergy Institute. Today, she's going to talk to us about all of the dirty details of soil microbial ecology and climate change. Thank you so much for joining us today.
Thank you for having me.
So first, in our kind of first vignette, we're going to go through a little bit about your system. So first, if you could tell us, what is microbial ecology?
So first of all, thank you for having me. So microbial ecology. Microbial ecology is the study of microbes in their natural environment. So our world is a microbial one. Microbes were here first. Maybe the first organisms on Earth. We all grew up sort of in this wash of microbes. Okay. And so a lot of microbiology and biology studies microbes in the lab, and our lab is really interested in studying microbes, sort of how they interact in their natural environment. So what are the things that change community structures? Why do some organisms show up at some places than others? Why does activity change? That's basically the idea.
Okay, so before we go any further, what the heck is a microbe?
Oh my gosh. So a microbe is any organism you can't see. Does everybody know that there are 3 domains of life? There's eukarya, there's all of us. There's archaea, which are the most sort of primitive forms of life, and the bacteria, and they are the most diverse by quite a lot. And so there are most of the eukarya, all of the archaea, and almost all of the bacteria you can't see. So if you need a microscope to see it, it's a microbe.
Perfect. Why should we care about these organisms?
Oh, wow. Okay. So microbes are everywhere and they do things in the environment. So for example, a lot of folks here are eating food, and our food first goes into our stomachs. That's where you get the nutrients, but you can't digest a lot of the kinds of nutrients that are in your food. We rely on microbes to break up fiber and make short-chain fatty acids, and those are essential nutrients. Soils, which we study, are microbial kind of superorganisms. They're alive. If you didn't have microbes in soil, it wouldn't be soil. The microbes make nutrients available for plants. They filter our water. People maybe think that trees are a source of oxygen in our atmosphere. That's true, but the major source is microbes in the ocean. So they're really kind of doing all of this kind of processing of our nutrients, and as well as making us sick and stuff like that.
Totally. So your work looks a little bit at the genome of microbes. Could you give us a little bit of a background on what that looks like in layman's terms?
Yeah, so all organisms have a genome. It's your DNA, and that's kind of your potential of the different kinds of things that you can do. And so I mentioned that the bacteria are super diverse. And so that means that they have a super diverse kind of set of things they can do. They can eat way more kinds of food. They can breathe way more kinds of things. Like we're limited to oxygen, and they can breathe manganese and iron and sugars and all of this other stuff. And so the genome tells you what is the potential, what kinds of things can an organism do.
So what does it mean to sequence a genome?
So when you sequence a genome, we extract the DNA from our organism, and we put it on an instrument that will tell us what are the different base pairs. So A's, G's, C's, and T's. Bacterial genomes are 1 to 10 million bases. Fun fact, our genomes are like 100 or 1,000 times larger, but we have the same number of genes. We all have about 2,000 to 4,000 genes. And so we get all those A's, G's, T's, and C's. So you can imagine like one sentence or like one word really that goes on for 10 million letters, all A's, G's, C's, and T's. So that's the sequencing part. If you're lucky, you get one word that's 10 million letters long. Sometimes you get multiple words. That's okay. And then we do something called annotation, where we take that one big word, that's the genome, and we kind of break it up and say, you know, where are the functional genes? What are the regulatory elements? What are other things that are going on? So that's kind of how we get the information about what organisms are doing, that annotation.
Cool. Very, very cool. So now we're going to zoom out just a little bit. How are our soils and the atmosphere connected? And how does that play into your work?
Yeah, so actually, I'm going to show a picture. I brought some slides.
Cool.
Okay, so this is a picture of the carbon cycle. Yeah, that's okay. You can still keep the captions. Yeah, so this is a picture of the carbon cycle. This shows, so it's terrestrial on the left and aquatic on the right. And this kind of shows where the carbon is moving. So all elements, carbon, nitrogen, even water, have cycles. They move through these reservoirs. And I'm interested in soil because it relates to the carbon cycle and the atmosphere. So you can see in the different areas, there are numbers. And those are the gigatons of carbon that are in each of those reservoirs. So for example, you can see that microbes respire a number that's like 60, so that's 60 gigatons per year. Plants respire about 60 gigatons per year, so that's CO2 going into the atmosphere, that's greenhouse gases. But plants are photosynthesizing that exact number. It's not a coincidence. So that they're in balance. The problem is this 9 gigatons of human emissions of CO2 that we're putting out through burning fossil fuels. So we're not going to solve the human emissions problem with our science, but we can better understand what are these microbial feedbacks to the climate system in the soil. And if climate warming is going to sort of change that balance of carbon going in through plants, sort of fixing carbon and carbon coming out through microbes eating food and respiring some carbon as CO2.
That is so cool. That is so cool. So you kind of mentioned that there are these reservoirs or ways that carbon can be stored. Could you talk a little bit more about what that means?
Yeah, so people think about the carbon cycle as sort of having these pools and fluxes. So soil's a major reservoir of carbon. Some carbon in the soil just doesn't get lost, as CO2. And this is an active area of research right now. And actually we have a new project trying to understand, why does some carbon stay in the soil and some leaves? And maybe it seems like, I don't know if it seems like you can't picture what it would look like for carbon to be in the soil, but I'm sure you can. Imagine a very rich soil that is dark and it smells wonderful. The darker a soil is, the more carbon it's storing. And so having plants growing in soil is one way to enrich carbon into the soil, because the plants encourage microbial growth and they fix carbon and put that in as root exudates.
That's awesome. So sorry, I didn't
No,
mean to
no.
cut you off.
What was the question?
Um,
Oh, you were asking me about
Carbon storage.
carbon storage in soil, or what it looks like for carbon to be stored in.
Yeah, like
soil.
just,
Yeah.
yeah, just kind of trying to get a sense of like how it works.
I mean, you could join my lab. You know, I think that's kind of what we're trying to understand is like, how does it work? There is carbon in certain places and we don't exactly understand why. I mean, some ideas are simple, like if you want more carbon in soil, you plant plants, maybe plant more native plants, maybe encourage them to grow, plant the right plant in the right place. More healthy soils tend to have plants growing in them, they have more microbes. But the question and kind of where we're coming in with our research is trying to understand, as the climate changes, some of these rules are changing in terms of why carbon gets stored in certain places than others. So a lot of labs are trying to understand this question.
Awesome. I think that's helpful. And I think it's also nice to kind of think about the fact that we as scientists, we don't have all the answers. That's part of why we're doing our job. So I think that was really helpful. How does the carbon that's stored in the soil end up affecting our climate?
Well, I mean, in one way, so soil is a possible like natural climate mitigation. So it's not going to be a solution. The solution is to stop burning fossil fuels. But storing more carbon in the soil will protect soil for all of the other kinds of good things that soils do. So sometimes people talk about natural things like soils as having properties that are called ecosystem services. I don't know if you've heard of that word, ecosystem services. It's a very kind of anthropomorphic, human-centered idea that like soils are doing things for us as people. We need special things, right? Microbes can live everywhere in the Arctic, in the bottom of the ocean. We have very specific requirements. We won't need pizza, and also it has to be hot. You know what I mean? Like, so part of what we are trying to do is understand, like, what are the ecosystem services that soils provide, and make those, sort of encourage those. So healthy soils that have a lot of carbon are not going to erode as easily. Healthy soils that have a lot of carbon are going to make plants that have more biomass and make more fruits and vegetables. Maybe they'll make more flowers to encourage pollinators and build up food webs. Things like that.
Yeah, okay. So one other thing that you're working on is you're studying some of the evolution of microbes. Something that we'll talk a little bit more about in the next vignette. But before we do that, how can you tell whether a microbe is evolving? And what does microbial evolution even look like?
We have tricks. We're using tricks to try and understand. So yeah, how do you know if an organism is evolving? So when an organism evolves, it changes its genetic composition in some kind of way so that it has a new function. Usually it's because it has a higher fitness, it can grow better, or it can make more babies. Just with microbes, it just makes more microbes. So we kind of take two approaches, which one is we look for evidence of adaptation, where we take a big collection of microbes, we question them for some kind of behavior, like can they eat cellulose? But then maybe we'll ask that of hundreds of organisms. And then we map those traits on a phylogenetic tree, And part of the way you can understand evolution is by kind of reconstructing ancestral traits using phylogeny. And so that's just it's just an idea that if you make a phylogenetic tree, that you can kind of infer what ancestral organisms would have looked like. It's just a model, it's just an idea. And then we use our genome sequencing to try and see if those changes were heritable and what they would have looked like.
Yeah, I think that's really helpful. So we're at the end of Vignette 1 at this point, and so now I'd like to open it up to our audience to see if they've got any questions for you. Yes.
So I'm sort of back when you said that we breathe oxygen, but microbes can breathe iron and other. So what does breathe mean in that sense?
Yeah, good question. Can I repeat the question for Zoom? So the gist of the question is talking earlier about how microbes can breathe things other than oxygen. What does that mean? How do they do that?
Yes, so such a good question. Thanks for asking. So when we breathe oxygen, it's called respiration. That's the actual scientific term. You inhale O2 into your lungs, and you have molecules or enzymes in your lungs that take that oxygen and put electrons on them. You get energy from that. That's your breathing oxygen. You're putting your extra electrons on the oxygen and reducing it. And so microbes can do that too, but they can put their extra electrons on oxygen, and that's very profitable. And most bacteria are able to use oxygen, but they can also use other things, like hydrogen sulfide or iron, and that's just putting the electrons onto something else. And it is very weird because those things are insoluble. So maybe they need to be touching them, or they need to have a pili that attaches to those things. Yeah.
Yes.
So is one of the things that you were studying about the evolution was what happens under the warming conditions? That's what I remember.
Yes.
Did you find an answer?
So the question is, what are you studying whether microbes
How they
evolve,
change.
how they change under
How they
warming,
change
and have you found the answer?
in a warming soil, not like Centralia. The person who's here before you see that, you know what Centralia is?
Yeah.
It's this coal burning fire in Pennsylvania
Oh,
that's been going for 25 years. So he used to go down and study warming soil there, but he left. So I didn't ever find out.
I think that this is what we were going to talk about next. Is the long-term warming
Yes,
study?
we're definitely going to talk more about it for sure. Hang tight.
I've been hanging tight for years.
We're so close to finding out. Yes.
Looking at your figure up here, it makes me wonder, how important is soil erosion to carbon flux on the globe?
Ooh, how important is soil erosion to carbon flux?
Well, first of all, this is the Department of Energy's idea of the carbon cycle. So it's not my idea. So how important is, well, it depends on your frame of reference. If you're a farmer or you're interested in the carbon cycling on your land, I would imagine erosion is a real big problem because you're losing your soil. And I know that soil erosion is a big problem in agricultural systems. In terms of the global carbon balance, I think it's probably a positive, because if the soil erodes, then it moves down into the bottom of lakes and oceans, and that carbon gets stuck down there. Because the deep ocean has quite a lot of carbon, and underneath the closed captioning there's like fossil carbon, but even subsurface, there's a ton of carbon down there. So burying carbon very deep in the soil is one of the more, how should we say, risky ideas maybe that people have in terms of mitigating this human carbon emissions. Because how, if you put something down into the ground, how do you know it's gonna stay there? It's very difficult to know.
Yes.
Yeah, as a farmer, we look at it as the carbon and the microbes create aggregates. So like little marbles that make it so water can infiltrate, and so you don't have that. And when it can infiltrate, it won't flood, which means it won't carry away our topsoil. And of course, then that yields to microbial necromass, which is the carbon that we're really looking for in the soil. But I have a question, because we've been finding out recently by Dr. James White about rhizophagy cycling of a plant, where it takes in, it ranches bacteria around the root tip and then sucks it into the root tip, dissolve the nutrients off of the shell, decides whether it's a type of bacteria it really wants to maintain or not, and then shoots it out the root hair. That's the purpose we found out of a root hair, and then reforms it again, grow it out again, and recycle it around, which is a major source of nutrient. What we're trying to understand, that I didn't know if we might, but we've heard that the plant can actually change some of the DNA of that microbe when it shoots it out. So when it grows up, it'll be more adapted to whatever resource that microbe's trying to take, whether it be zinc or boron or whatever. And the exudate it feeds it can then make it more compatible. But I don't know if you know
Sounds
anything
good.
about
I don't, what is it called?
rhizophagy cycling. Yeah, Dr. James White out of Rutgers figured out about maybe 4 or 5 years ago now. And farmers have gotten, because then we can really focus on keeping that microbial area around the roots, really healthy and structured, keep that cycling going, which can reduce our nutrients and mineral use by a tremendous amount.
Great. Well, thank you all so much. With that, we're going to go ahead and move into our second vignette, and we're going to dig a little deeper, if you'll pardon my pun, into some of your work and what you're doing. So if you could talk just in kind of a general overview sense, what is the focus of your lab and your personal research?
So, so the purpose of our lab is we're interested in the microbiology of climate change. How is climate affecting microbes, and how can we kind of use that understanding to try and make some tools like biofuels or microbes that can improve biofuels to help mitigate climate change.
And you have some very hot off the press research that just came out this month.
That's right.
That's kind of about some sort of microbe adaptation to warming climates. Could you give us a little bit of a summary on that?
Yeah, so let me, maybe I'll start at the beginning, since we have time. So the picture that's on the screen now is of the Harvard Forest, which is in Peterson, Massachusetts, just right off of the Quabbin Reservoir. And this 4,000 acre experimental forest, it's like a picture like a national park, but it's set aside for research instead of for recreation, although it is open for recreation, and anybody can go out to the Harvard Forest and hike around, and they have a wonderful museum, and you can walk by all of these experiments that are happening out there. And so I came to UMass in 2011. And when I came to UMass in 2011, I was introduced to Jerry Melillo and Srita Fry, who had set up these experiments. I'm not a field biologist, but I love working with them. And so they set up this field experiment. It's a long-term warming study where the idea is they wanted to sort of look into the future of climate change to try and understand, like, what are soils going to look like if we were living in, say, a 5 degrees above ambient trajectory. At the time, this was a worst case scenario. We are cruising towards 5 degrees at a clip that is pretty frightening. And soils especially, one of the things that they do is they serve as sort of a buffer. And so the soil temperature doesn't change as much as the air temperature. So 5 degrees of warming in soil is quite extreme. You can see maybe there are 2 experiments. There are these large plots that are on the left and then small plots on the right. So the large plots on the left, that's the warming study that was part of this Mallory Shador's paper that I shared with you. This is experiment has warming treatment. So there are these 6 meter by 6 meter plots. There are 6 replicates of them, and they're heated through these buried heated resistance cables. I don't know if anybody here watches football. Did you ever see a football game on TV? And like maybe the Green Bay Packers are playing and snow is like driving down and it's collecting on everybody's hats and the field is green. This is the same technology. Actually, Jerry was he was not a professional football player, but so this is the same technology. There are these heating cables that are buried 10 centimeters deep, and every, I don't remember, like 10 centimeters or so, there's something called a thermistor, and that's just a fancy word for a thermometer that's connected to a computer. And so there's a shed with a computer and a data logger, and in some of the later pictures, you'll see the shed where all of the electrical is. And every 5 minutes, this computer takes the temperature of the control plots, which have these buried cables, but they're not heated, and then puts enough resistance through the heated plots to heat them 5 degrees above ambient, whatever ambient is. This has been running since 1991. I know, so cool. I was in high school. Like I wasn't even thinking, I didn't know what a microbe was in high school. So I love that I get to work here. It's such a privilege. And so what Jerry's group has been doing, and now Sarita's group, is kind of following this trajectory of what is happening in the soil over this amount of time. So this picture shows a close-up of one of the plots. There are 6 replicate heated plots. And maybe you can see in the background, so that, do I have a pointer too?
I think that has a pointer on it.
See if it works. I want to press to get.
Oh,
wait, there it is. Okay, so this is a heated plot, right? You can see the outline. And then this is a control plot. It still has snow. So I like this because it kind of gives you a sense of the scale of this. We don't sample in winter though, the ground is frozen. So, but I like this because you can really see the effect of the warming.
Yeah, absolutely. So what types of differences did you then observe between that heated soil type and the control soil microbes?
So when I got to UMass, it was 2011 and it had been like 20 years. And Jerry and Sarita were noticing that the soils were just hemorrhaging carbon. So a lot more CO2 coming out of these heated plots than the control. And I know there's a bunch of scientists in the crowd. And so you know that like if you heat any biological system, the metabolism is just going to go a little faster. So that's not that unexpected. What was strange and what got me involved was that this carbon loss was not linear. It wasn't like they heated and then every year there was more and more. They heated it in 1991, and for about 8 years, every year there was more. And then for like another 8 years, there was no more treatment effect. Right, this was Jerry's face. How could there be no treatment effect? And so then they started trying to think of like what could be happening, that we're heating these soils, the kinetic effect isn't working anymore. So they came up with some ideas about like maybe all the food is gone, the food for the microbes. Maybe they've adapted, and that was true, that the microbial communities changed. But if all the food was gone, how did that explain phase 3 when the heating caused sort of another burst of carbon loss for another like 6 years? And so that's when we started thinking like something's going on where maybe the microbes are stressed And then these periods when it's not, where there's not this warming effect, is some kind of change happening in the microbial community. And it was sort of having these thoughts is when I came into the project. I was a new assistant professor and I was looking for something fun. And this is a great group of people in the Harvard Forest, just like a wonderful resource, a lot of great scientists, a great network. And so we started working on this project and trying to understand. So, the short story is these soils over 30 years have lost 1/3 of their carbon, 1/3. It's really nuts. And so there's a lot less carbon because they have so much less carbon. They don't hold water as well. So they experience drought more often. It's also changed just how they look. I don't know if you've ever dug a hole in a forest around here, but there's this really thick organic layer. It's like a rug, like a mat. And you can actually kind of lift the whole thing up. It's like a net that's connected of fine roots and fungal hyphae and decaying leaf litter. And then underneath that is the mineral soil. And that's where the soil minerals are and all that other organics. There's microbes with all of that. And so this organic horizon has shrunk. And in some parts of these warming plots, they're actually bare. Does anyone have the pleasure of visiting a tropical rainforest? Highly recommend. But if you look on the ground in a tropical rainforest, there's no organic layer. The leaf litter decomposes so fast that you see the mineral surface on the ground looks just red all around. And that's what it's starting to look like in patches. We're just losing this organic layer. So the first thing we did was we looked at the community ecology. We thought, let's see if the communities have changed and maybe there's less microbes. Well, the communities changed a lot for the fungi and not a lot for the bacteria. And the biomass decreased a lot for the fungi and not at all for the bacteria. And so that's when I started thinking like, well, if there's the same number of microbes and the environment is so different, and seems like it's more stressful, there's less food, there's less water, like that's pretty objectively more stressful, even for bacteria, which can do all kinds of things. So that's when we thought we should start looking at adaptation.
Wow, that's amazing. It's so interesting. So you kind of looked at the genomic signatures of all of these bacteria. What kinds of responses do you expect? Another 30 years into the future, 100 years into the future, what do you think this trend is going to show us?
Yeah, that's a good question. I'm not exactly sure. I mean, I can tell you what happened in 30 years of warming, because that's kind of our look into the future. This is the big idea, is that we're warming these for 30 years with the idea that maybe in 30 years, the control soil will look something like this. And that's been the interesting challenge, actually, of doing this. So, over this 30 years of warming, our baseline is also moving. And so we're always having to kind of compare because the landscape is shifting. But so far, what it looks like is the bacteria in the heated plots are better able to decompose the carbon that is left. And so that makes sense, right? They've improved their capacity to degrade what little is left. We have an undergraduate right now who's looking at this question about competition. So if there's less food, are microbes competing more for what's remaining? Maybe making more antibiotics or having more competition. So that's something that we don't know yet. We looked at how they grow in relation to temperature. And interestingly, their temperature sensitivity, this is another undergraduate project, person who just graduated. The temperature sensitivity did not change. This is something that was noticed for the communities, that the community temperature sensitivity did change. So I mean by temperature sensitivity is, I told you if you warm a system, the rate of respiration increases. So if you measure how much it increases, that's the temperature sensitivity. And so in the communities and in the soil, the temperature sensitivity is lower for communities. So you heat them up and they don't change as much. But for the individual bacteria, we did not see a change. Their temperature optima is higher. So they prefer a higher temperature, even though how they respond to temperature in their growth doesn't change.
That is so cool.
Yeah.
That is so cool. So to kind of wrap up this vignette, this research, as you kind of mentioned, took place over several decades at this long-term research site. Can you speak to the importance of doing long-term research at sites like this and how the results from studies like these might differ from some of the shorter-term studies that we tend to do?
Oh, well, I mean, this is kind of a, well, it's not a completely unique resource, but there are not a lot of resources like this. Something I didn't mention, but our work is supported by, in part by the National Science Foundation, and they have this program called Long-Term Ecological Research, LTER, and there are, I don't know how many, something like 15 maybe LTERs in the country, and Harvard Forest is one of them. And it's this funding program specifically intended to support this kind of long-term study, because there are just some things that you can't understand in a shorter time frame. So I do periodically go to Washington, D.C. and talk to my representatives and senators about the importance of funding this kind of long-term research and what a wonderful resource it is, not just for science, but for training students and building up our networks and our sort of technological ability to understand these systems in the first place.
That's awesome. That's so cool. Okay, so to kind of wrap up our second vignette, we'll open our questions again to the audience. If you guys have any more questions about this section, we'd be happy to take them. Yeah, Janelle.
I have one from Zoom. They said that if the heating tables that you talked about, they're buried and only heating the soil, does that accurately reflect what our like ambient warming is actually going to be? Because wouldn't the air temperature also have impacts on what's happening to the soil?
Yeah, ooh, that's a really good question. By saying that work, but we're all just people doing our best. This is just what we can do. It's a limitation. And actually there are 3 warming experiments happening, and one of them is shutting down this summer because they're hard to run, they're expensive, even though we're still only looking at you know, the below ground. One of the other, like, we're only heating partial root areas, so we only look in soil. We can't really understand trees or larger organisms. We can't look at insects because they can migrate. It definitely has its limitations. This is where working in the lab can be useful, but then in the complete other end of the spectrum, the Department of Energy, which has funded some other of our work, has also supported this other project called Spruce. And I think Spruce might be an acronym, and I don't remember what it stands for. But Spruce is a warming experiment, kind of like Harvard Forest, but they're heating underground, and they've built these giant 60-foot high greenhouses around stands of trees. It is bananas. And so they're heating whole stands of trees and underground to try and understand this question. It's a huge endeavor. It's like the CERN of ecology. It's nuts.
Wow,
Yeah.
That's amazing.
So yeah, it's a caveat of our work. Definitely.
Cool. Definitely.
Where is it and how long has it been in effect, the Spruce project?
Spruce. Spruce is in Washington State, and it's been running for like 8 years. That's a guess. Does anybody know? Yeah, something like that. It's newer. Yeah.
Yes.
So you mentioned that carbon-rich soils hold water better. And my question is, why is that? Is it related to the less organic matter in that pool that you were talking about? Why do carbon-rich soils hold water?
Ooh, I forgot to repeat the question for Zoom, but why do carbon-rich soils hold water better?
So more carbon-rich soils have more organic matter, and it just acts like a sponge, yeah. So it's partly organic matter, and I know we have some soil people in the room, so it's partly clays. Clays have layers, and the water can store in between the layers. And so that might be another reason that if you have more water, the layers can expand and contract. But the organic matter, it just sticks to it.
Following up on that earlier question, I mean, another thing you can't mimic so well at Harvard Forest is, that I assume they did not raise the temperature in a gradual fashion, which is what we're actually seeing. So the microbes don't have a long length of time to adapt. So I assume it was a sudden 5-degree.
Yeah,
They just turned it on.
flip the switch.
So I'm wondering how significant you think that might be in the microbes response.
Yeah, so how significant is it to have a fast temperature switch instead of a slow gradual one?
Yeah, that's a good question. But it's hard to answer, without doing an experiment. I mean, what I can say is, I know that there are lots of studies doing this kind of warming, like in this experiment, we're heating through these buried resistance cables. People will sometimes put up passive heating elements, like, what are they called? Like the little glass houses. I'm blanking on what they're called, but it just looks like a little greenhouse with an open top chamber. And that will keep the soil like a degree or two. There are also, sometimes people will hang these heaters to sort of radiate heat to the ground. And those two methods heat less. And then you sometimes just don't see as much of an effect. I mean, we're heating 5 degrees. And the temperature range that the soils experience between winter and summer is between maybe -10 and 25 degrees. So there's a 35-degree range, and we're heating only 5. Just to give like a context of what that's like. Yeah. So you're right. I mean, the whole thing is unnatural. But maybe, yeah, be fossil fuels are too.
Yeah, just thinking about his question, how long do the microbes live, the individual microbes?
How long do microbes live?
Such a good
So
question.
is it new microbes all the time? That's what I'm thinking.
Yeah, it is new microbes all the time. So we have like a community average measurement of how often a microbe lives. So how long a microbe lives, it's a little complicated, right? Because when a microbe is alive, it divides. Now it has 2 daughter cells, and the original cell is not there anymore, kind of in the new cell. And so what we measure is that it's every 2 weeks. That's a community average. So on average, every 2 weeks, there's a whole new set of microbes.
Except they're not new because they're
They've divided, yeah.
But then if they divide too much, the contribution of the original microbe gets smaller and smaller, doesn't it?
Well, there's no original microbe. It's new.
Well,
I know. Yeah. So I want you to think about another idea. And this came from Melissa, who's here, a good idea. So we talked a little bit, Ed brought up about necromass. I don't know if people know what this is, but I'd like to explain it just because it's a cool idea. So I told you that over this experiment, the number of microbes doesn't really change. So imagine over the course of a season, right? We're just getting into our growing season. April, we'll go right into November. Every 2 weeks, we got the microbes that are there are doubling. But over the course of the season, the total number doesn't change. So what's happening to half the microbes every 2 weeks? They're dying either through predation, or they're lysing, or they get a viral infection, or they run out of food. And they become the new organic matter.
can they get eaten?
Yeah, they
Oh,
can.
that's predation.
Yeah, that's predation. Sorry. Yeah, they get eaten. And so that's partly what is the new organic matter. And so this idea that microbes are the soil organic matter, not like undecomposed plant material, is a relatively new one to science. Maybe in the last 10 years, people have appreciated that dead microbes are building soil.
But what about the fungus that went away? Didn't you say that the fungus
Yeah.
material disappeared more?
The fungal biomass is lower.
So did the microbes eat it?
The dead fungi, probably some of them, yeah. Yeah, oh, I love Last of Us, I just ate that vegetable all up. Delicious.
So good.
Is there any plant cover on, ground cover on either of those two plots, or has that changed over time? As far as like feeding exudates to keep part of the community going?
Yeah, there's good ground cover in all the plots, I mean,
Oh,
we don't
okay.
plant anything, it's
Right,
just. the
but
natural-
natural, but you do have some. So that's something
Yeah,
you.
there's plants,
Okay.
yeah.
But you'd expect the heat would encourage the plant cover. Right? On certain times of the year anyways.
Yeah, well, it's definitely extending the growing season,
Yeah.
and that's what we're
Yeah.
seeing in our natural climate change progression as well, yeah.
Janelle, yeah.
I just have one quick question from Zoom. Are you able to actually monitor if the species of the bacteria is changing over the time?
Ooh, you
What
wonder
species
how
are
the
they?
species of bacteria is changing.
So we can, the species definition for microbes is a little contentious, I guess. But we can assign species, and that's one of the things that we do with sequencing. Is there are some genes that everybody has, me and you and our mitochondria and all the bacteria and archaea. And those genes are unique, and we can sequence those genes and kind of assign a species name. It's a taxonomy, just say what it is. And so when I was saying that the community change was different, that's what I meant, like species of fungi were different and the species of bacteria weren't that different. And it matters because more closely related organisms tend to share more functions. And so we do community ecology, not because we care about, Fred or Margaret or whatever the microbes names are, but because they do certain things. And so yeah, that's something that we do.
Awesome. Okay, so with that, we're gonna move into our final vignette of the evening. So how did you start working on climate change and microbial ecology? And what kind of inspired you to start down this path?
You're dread. So, yeah, it's a good question. So, like, I discovered microbiology first. My students have heard the story. It's like process of elimination. I was just looking for something to do and I liked science. And I discovered microbiology and I thought they were really cool. And that was my thing. And then it was about that same time that I kind of like woke up to climate change and sort of realized that this is not, this is something I should pay attention to. It's something that's affecting everybody, and it's going to affect all of our social systems and ecosystems. And I spent a long time trying to figure out how I could study microbes, which I love, and climate change, which I care about. And at the time I was a postdoc, like thinking about these things, and I was working in UC Berkeley, but we had field sites and collaborators in Puerto Rico, and I was flying to Puerto Rico. I'm feeling awful about my carbon footprint, and I'm actually from Massachusetts. I grew up in Watertown. My parents still live there. They might be watching. And so I really wanted to come back to Massachusetts. I always wanted to live here. And then when I came to visit UMass, I got to visit Harbor Forest, and we have this wonderful LTER, like only 30 miles away. I thought, this is amazing. And so it's just like this kind of confluence of like, what I'm interested in and what I want to do. And, soils are so special and they need protecting. And actually, I'm gonna be a little bit of a stickler for a moment, because well, these are the dirty details. Dirt is soil where it does not belong. There, I said it. Nah, we can all relax.
Do you have any advice for any individual that's interested in this topic, or suggestions for any ways that they could find some opportunities?
For people who are interested in this topic. So, I mean, I'm pretty passionate about soil conservation, and that it's a real like personal responsibility. And I think everybody has some soil in their life that they could take better care of. So, you know, here's a few things like having a lawn isn't the worst thing. It's not the best, but if you really want to have a lawn, maybe don't cut it so short. Or maybe set aside part of your lawn and plant some wildflowers so that you can encourage pollinators. Planting anything at any time is good. Don't dig up your, you know, invasive plants or your non-native plants to plant native ones. There was a great study by, I think it was Bethany Bradley's group a few years ago that said 70% is the number. If you have something like 70% natives, that's still, you're doing fine. So there's a lot of ornamentals that aren't native, they're fine. So that's what I would say, like, take care of your soil. Don't have bare patches, plant some flowers, maybe don't mow the whole thing, have a garden, like take care of your space.
Compost.
And composting, yeah. I would love for Amherst to have municipal composting.
Those are working on it.
Yeah, that would be great. I'll look forward to my compost that I get delivered.
And what would you say for new students that are interested in kind of taking the path you took?
My path was super non-linear. So I don't know. I mean, and a lot of it was luck, and a lot of it was just kind of being patient with like the fact that I didn't have my acts together for a really long time. So I don't know if it's hard to give advice because I'm not sure I would advise to do exactly what I did. But I took a lot of years off in between everything actually. And I talked to a lot of different people. And so that's probably good advice, is just talk to a lot of different people, especially you students. Graduate students and undergraduate students, every door on campus is open for you. People are waiting literally to talk to you. All you have to do is ask. And so I know it's like you feel hard and you're like, feel like you're bothering them. You're not bothering anybody. Like, we want to talk to you. People want to talk to you. It changes once you graduate. As a postdoc, and then as you get older, it becomes more your responsibility to mentor. But while you're a student, now is the time. You can send any email to anybody and ask any question. And so you should take advantage of that. And that was what I did. When I was looking for graduate programs, I probably sent 100 letters to people. Just, hi, your science looks cool. Can you talk to me? And like, most people ignore me, that's fine. But like, I learned a lot, and that's how I sort of ended up here.
Yeah, I think that's such valuable advice, because it can seem very intimidating to go knock on all those doors as a student, but that is so valuable. So switching gears just a little bit, although the evidence of climate change is all around us, there's many of us that don't work directly with climate change, but we still kind of feel this anxiety about it. Your lab is in very close proximity to the issue, and we were wondering if your lab group stress and some of the ways that you let off some of that stress.
That's a good question. I mean, I personally struggle with climate-related stress, and I'm not sure. I don't exactly know how to deal with. I mean, I think part of the way that I deal with it is I changed my whole life so that I could work on climate change. You know, that was one way. I do some volunteer work, and that helps me feel better about climate change. I've done some get out the vote work with the 500 women scientists, which we were talking about, and I work with the MSP union, so it's a Mass Society of Professors, is sort of helped to organize this multi-union initiative. So that's like PSU and GEO and the undergrads and with Sunrise and a bunch of different groups. We have this group called ESAM, which is Environmental Social Action Movement. And the idea is this is the new like climate justice committee. But instead we're a group, we use something called sociocracy, which is just a way of sort of distributed group decision-making through consensus and conversation. And we work on things like voting is the circle I'm involved in right now. We're also working on transportation and trying to make transportation more green. We're working on, I think plastics on campus is on hold. We're working on banking. That group is very active, and you've probably heard about some of the work they're doing to try and get the sort of fossil fuel enabling banks off of campus and get campus to do more to sort of divest their financial portfolio from some of this fossil fuel emission stuff. And then we do a bit of outreach too. So that's been really actually cool, like good way of feeling like I'm not just, I don't know, I definitely get the feeling sometimes, I don't know if my students do, of like, you know, I'm studying microbes because they're cool and there's all of this stuff happening in the world and maybe I feel a bit disconnected. So, I mean, we're thinking about it every day and I feel disconnected. So I think those feelings are unavoidable. And then maybe part of it is just like the best sort of way of dealing with it is just to have a good time anyways.
I love that. I love that. We talked about this briefly earlier, but I wanted to touch on it again. Some of your research has been funded by the Department of Energy. You mentioned the National Science Foundation, which means that in part you're funded through taxpayer dollars, which is a great way to get science funded. But can you talk a little bit more about what being federally funded means as a scientist, and what that entails, and how that kind of overlaps with your own mission and goals?
Yeah, that's a good question. So our lab is supported only through federal funding right now. So it's Department of Energy, the National Science Foundation. And the way it works is a little bit different for the two. National Science Foundation, their mission is research and training. And so, you know, I mentioned that some of my students are here and there was a picture of some of the students, like dozens of people, not just me, have been working on this and have contributed to thinking of new ideas and developing them and actually physically doing the work, writing our papers, mentoring younger students. And so that's the National Science Foundation. And then Department of Energy has a little bit more of a specific mission, or at least those programs have more of a specific mission. But for both programs, the idea is that, you know, we come up with a research topic that we're interested in. We write it up as a grant proposal and talk about not just what we're going to do, but how we're going to know if it works and who's going to do it and how are those folks going to be supported. And what are the things that we're going to do to make sure that everybody knows about this work that we do, not just publishing, but going to professional meetings and coming to things like this and talking to high schools and middle school students. And in fact, you know, we get these budgets and about half, a little more than half, goes to the university. It's called overhead. And it's just, you know, it pays like janitorial staff and facilities and all of that. But then the rest of it that I get, almost all of that goes to support students and postdocs and undergrad workers in the lab. So a lot of that money, I mean, it looks like science, but a lot of it really is new scientists.
That's awesome. So our next question is, tomorrow is Trans Day of Visibility. Could you possibly share the work of some trans scientists that you admire
Yeah. Yeah, so there are two people who are out and trans, who are scientists who I want to mention. And there's not a lot of out and trans scientists, and I know that people are discovering their sexuality and gender identity all the time, but so I'll talk about two. So one is named Dr. Micah Tosca. I don't know if anybody was at AGU and saw Dr. Tosca's talk. Dr. Tosca is a climate scientist and artist, and her work specifically explores like how art can help in science and science communication. And she's a wonderful science communicator, and she's at the College of the Chicago Art Institute. I didn't even know they had a college. Definitely want to work there. So I would say that that's one person, because I love this intersection of art and science to kind of use art as a way of conveying ideas or making science seem more relatable. And then the other person I want to mention is a new member of our UMass community. And her name is Dr. Anna Marie Lachance, and she's in chemical engineering, and she's a new lecturer, and we haven't actually met yet, but we're having coffee next week. You're watching Anna. And we share a lot of the same passions in terms of expanding access for learning, to broaden the ability for people to learn in lots of different communities. Trans rights are human rights. Science is something that should be for everybody. And anyway, those are two people that I like.
Wonderful, wonderful. With that concludes the end of vignette 3. So if there are any more audience questions, we'd love to take them.
Yeah, I have one from Zoom. Do you have any advice for folks who have environmental backgrounds, but want to find a way to get into this type of research without going back to grad school?
Ooh, so do you have recommendations for people that are in environmental work to keep going but not go back to grad school?
That's a good question. Let me think about that. You know, there are resources available through our societies. So I know in particular, like Ecological Society of America has professional resources, like certificate programs, and American Society for Microbiology, ASM, also has resources on their website. So I'd probably start with our professional societies. In terms of environmental science, the only other thing I can think of is I got involved a little bit with the Ecological Landscape Alliance. I've lost track of them through the pandemic, but that's a local group who's made up of land managers and land stewards and people who are sort of in charge of environmental decisions. And they're really passionate about science and ecology. And they have a lot of great learning materials on their website. They have a newsletter, they have a conference. So those are some resources that I know about.
Anything like citizen science? Are there citizen science projects that people can get involved in?
Ooh, are there citizen science projects that people can get involved with?
Wish I had one to tell you, but I don't know of one. I'm sure that there are. Do you know? Oh, yeah.
Just a coincidence. I was looking at a citizen science project yesterday that involves iNaturalist, which is an app like training for identification, valorization of the wildlife we have here. And they're having actually this month of April, a project in partnership with the Mount Grace. I forget the full name, but the reservation. So they're gonna have nature walks to train people on how to use this and contribute to the generation of data through citizen science, and also have these nature walks through public and private property to look at the wildlife that comes out during the spring. So that's something that would be cool to check out.
Was that through the UMass libraries?
Yes.
it was an email from the UMass.
And it was an email from the UMass Libraries, but I think the program itself is.
Yeah.
Yeah.
Actually, I'd love to also direct people back to the Harvard Forest. They have a big education component. And so there are resources on their website, there are public talks.
So there's also there's that one section of the National Science Institute, National Science Foundation, and that is devoted to broadening impact. And so there must be people who know about many different fields that have citizen science type projects. Remember Julian, whatever his name, with the rice, the arsenic rice? You don't remember this? What was Julian's name? He's a physicist. But I know I did one of the experiments when my daughter finished. She said, "Oh, thanks, Mom. Put some arsenic in my pots." But he had people measuring the arsenic in rice and sending it in electronically, and then they would use the research. Tyson. Really.
Oh,
Yeah. And I was so glad you gave such a big natural boost to water.
We noticed that you have plankton on your website from SpongeBob. And we were wondering, we included that in the poster, and we're wondering why you have plankton on your website.
Yeah, so what's your connection to plankton from SpongeBob?
well, I mean, SpongeBob is amazing. Yes. And hilarious. I don't know of another sort of microbe that's also a comedy giant.
Yeah.
I don't think there's another one. I'd love to know if there is. But also, I find plankton kind of hilarious because he's constantly failing, even though his plots are nefarious, because he's acting alone. And microbes are meant to live in communities.
Yes, I have a question about microplastics in soil. And we hear so much about it, and there are certain microbes that can eat plastic that are going to save us in the future.
Well, so the question is, what about microplastics in soil? And is there any way that microbes could potentially eat them in the future?
So there are plastics in soil and everywhere. There are microbes that can eat the plastics, but as far as I know, there aren't a lot of microbes eating plastics in soil because they have better things to eat. So plastics do break down, but part of that decomposition is called like abiotic, like it's through UV degradation or physical shearing of the pieces of plastic, not so much microbes eating it. I know that some people are working on trying to sort of engineer microbes to eat more plastics, it's a tricky kind of science because even just finding the plastics is difficult if you were to sort of go out. I mean, I don't know. I've bought compost locally and found plastic in it. I don't know if you can buy compost without plastic in it. It's just everywhere. So I hope that we can figure out a way of composting plastics to get rid of them, but the bad news is plastics now are starting to lithify. People are finding rocks made of plastic.
It sounds like a good use of, good way to get rid of.
It just makes them harder to get rid of. I don't know. Yeah, maybe they're less reactive.
They're so non-nutritious.
I know, that's why it's hard to get microbes to eat them.
Right. And if they did eat them, they die, right?
Well, there's a microbe to eat everything. They might not want to eat it. Might have to force them to eat it.
You wouldn't want them to go wild. And all of a sudden, your siding on your house is disappearing.
Oh, yeah.
just before we go, if you would mind, if you had to like summarize the most significant findings from your Harvard study in like one statement. what would it be?
Ooh, if you had to summarize the most important findings in your Harvard study, what would it be?
Okay. So the long-term warming is causing this loss of soil carbon by a third. But the microbes seem to be adapting to lessen the decrease of carbon over climate change time. So this adaptation that we're talking about, this evolution of microbes and what they can do, seems to be kind of swinging in this direction of mitigating part of the problem. So that's a good news.
Okay, with that, we would like to thank you again so much for being with us. This was such an awesome,
you. It's my pleasure.
awesome conversation. And thank you guys all for joining us as well. Look for emails or on our social media about our next event, which will be next month with Dr. Jess Bersoni here at Amherst Women's Club, as well as virtually on Zoom. You can also check out our website for more information about all of our various events. And if you haven't already, please don't forget to fill out either the virtual sign-in sheet or the sign-in sheet that we've got kind of over there at the front of the room, so that we can continue to give you guys updates on what we're doing next. And with that, have a great evening, everybody.